Yes, you may start whenever you are comfortable.
Good evening, ma'am.
Good evening, Siddesh. How are you?
Ma'am, I'm doing good. Just finished the presentation and now just opened it in PowerPoint mode.
Okay. Start with the introduction of your group first, okay?
Yes, ma'am. So, hello. So, I will introduce the members of the group. So, we have in our group Gayatri, Purvi, Samin, Paripurna, then Vaidehi, and myself. And we together did the project, dividing all the tasks between us. So we individually took each task, and today Samin and me will be presenting the presentation of how the project was executed using the Biopython code which Muskan ma'am taught us.
Okay, who will be sharing the screen?
I will share.
Okay.
Is it visible, ma'am?
Yes, it's visible.
Okay. So may I start with the presentation?
Yes, you may start.
Okay. So hello everyone. Today I'm going to start with the presentation of a project using PowerPoint presentation. So here I'll uh, first introduce the topic of our project, which was in silico identification and functional characterization of one of the antigens from Oriensia tsutsugamushi, which is a bacterium, using sequence analysis and homology based annotations. Uh, we were together six members in the group and we did the project. We were guided by Muskan ma'am. So I'll first introduce the disease and the pathogen. So the pathogen is called Oriensia tsutsugamushi, whose visual representation as a cartoon can be seen on the right hand side. It is a gram-negative obligate intracellular bacterium. The structural rigidity to the wall is conveyed through cross-linked outer membrane proteins. They are known as type-specific antigens, and they are of different sizes. And of our interest is the protein 47 kilodalton size, that is TSA 47. The disease is called scrub typhus. It is an emerging neglected tropical disease which causes about 1 million cases per year. It is endemic to the Asia Pacific region, whose visual representation can be seen on the right hand side, called Susugamushi Triangle, and India is one of the affected regions as well. The transmission occurs by a larvae of harvest mites, which are also known as chiggers, whose cartoon can be seen on the right hand side. So it acts as a vector.
Siddhesh, you are mute.
Sorry, ma'am. May I continue from this step or should I go for the earlier step?
It's okay, continue with this.
Okay. So I'll briefly introduce the protein itself. So it is a trimeric protein, which is TSA47 present on the surface of the bacterium. It is highly conserved between different strains of Orientia and is known to be involved in budding out of host cells. It is highly understudied regarding its role in Orientia's life cycle, which is why our objective was to dissect it in silico and extrapolate the function of this protein using Biopython. So now I will continue with the step one, which was sequence retrieval. These are the respective protein IDs and the nucleotide IDs. This was the input syntax, where I have also described why and what did I exactly do in this code. And this was the output of the file. So you can see we have successfully got the output, which gives us the length of the sequence and other details, including the sequence itself. So, which is why the protein from Orientia is shown to have 466 amino acids from the code. Step 2 was sequence quality analysis. So I couldn't fit the whole code in one PowerPoint presentation. Anyway, this will be there in the GitHub, but this is just to briefly show how it was done. And this is the part 2 of the input syntax for the code. So from this, um, we also get the output successfully, and it shows that all individual amino acids could be enumerated within the protein. And I also mentioned the percentages of different types of amino acids. The reason for mentioning this percentage of different types of amino acids here is because, as we know with the Ramachandran plot, that it affects the structure of the protein, and structure and function are linked together. So, for example, the proline occurs I mean, amino acids like tryptophan have big groups, so they affect the protein by giving it steric hindrance. So, next part of the sequence quality analysis was to check the quality of sequence using sequence length, low quality basis, N-terminal and C-terminal residues, and also to check the internal stop codon. I would like to briefly mention that for this particular step, so Gayatri did the code. Uh, so we can see the code part here. And as one can see that it has ran successfully, which means the sequence passes the criteria, all the 4 criteria, which is why the query sequence was further used for analysis. And after this, for the step 3 and 4 and 5, I would like to hand over the presentation to my colleague Samin.
Ma'am or and Sadesh, is it okay if I screen share?
Yeah, I will stop sharing the screen now.
Okay. Is my screen visible?
Yes, it's visible.
Yes.
Okay. Okay, so good evening, ma'am. My name is Samin Khalid, and I'll be continuing this pipeline from step 3 till step 5. So basically, in this pipeline, we performed sequence filtering. In this step, the sequence filtering and validation. The main purpose was quality control. So basically, we initiated that by firstly, the CDS region were extracted from the GenBank file using Biopython, and at the DNA level, the parameters were selected, such as sequences shorter than 90 base pairs were removed, valid nucleotides were only considered, and length of those sequences should be divisible by 3, and the sequence must start with ATG or start codon. At the protein level filtering, the parameters were that there should be valid amino acids only. There should not be any internal stop codons, and stop codons should only be allowed at the end. So at the end, we get two things. Number one, we got a filtered DNA sequence, and then we also got a filtered protein. So the filtered FASTA file was a quality check pass protein, and it was a TSA47 protein consisting of 466 amino acid. So this is the command section, and you can see at the here that the high quality protein file was saved. I have also added this filtered DNA sequence, and this is the filtered protein sequence. Now coming towards the next step, which is the homology search using BLAST. Our purpose was to identify two basic things. Number one, similarity search using BLAST, and number two, closest homologs, conserved regions, and evolutionary hints. So for similarity search, just as we discussed in the step 3, we were using a filtered protein sequence file, and a BLAST-free similarity search was performed against the RefSeq protein database. BLAST was performed successfully, and the results were saved in an XML file. For homology, okay, so you can look at this. This is a snippet of how the XML file looked. So for homology search, our objective was to identify the similar proteins from Orantia Sutsugamushi, and the method we used during Biopython included the parameters that were telling us about the homolog length, e-value, HSP score, identity, query region, and subject region. So from this result, the total alignments that we got, all the similar sequences we got were 50, and the top hits showed 98 to 100% sequence identity with e-value that was zero point 0.0, and full length alignment from query region 1 amino acid till 466 amino acids. So we can interpret that extremely low e-values and high identity is indicating the strong homology. It also confirms the correct identification of target protein. So this was the command section, and these were the top 10 closest homologs. As we can see that from 98% to 100% is the identity or score of all these top homologs, and we can also see that e-value remains 0 point 0.0, which actually indicates that these are the strongest closest homologs. So the functional annotation from homology was thus that, as we can see in this, that all our proteins were DECP-like serine protease TSA47, which shows that this protein is highly conserved. So for the conserved region analysis, we also perform, as I've shown that this is how we find the conserved regions using BioPython. So from the conserved regions, we understand that there are several highly conserved amino acids, for which actually means that from this position of these amino acids or the alignment of these amino acids are conserved among all those homologs that we have found. in closest homolog section. So these conserved regions could possibly suggest their functional importance because these positions are not changing, which are actually telling us that how structurally stable this protein is and what could be the possible role of this protein in host pathogen interaction. Also, for the evolutionary insight, we also did a command here for evolutionary hints that what is the possible evolutionary analysis of this protein. So observation was that all closest homologs belong to Orantia Tsutsugamushi, and there was no distant species that were detected. So what we can interpret that TSA47 is a species specific, which technically means that it is only found in Orantia Tsutsugamushi, and it all shows low evolutionary divergence. So basically, in this insight, we can, infer that homology-based analysis it confirms that TSA47 is a highly conserved, functionally important protein in Orantia Tsutsugamushi. So coming towards the step 5, which is the functional annotation, as we have discussed that it's that TSA47 is a serine protease and its purpose is housekeeping the exported proteins, and its role is in stress resistance during active exponential growth. So the basic purpose is of basic purpose of functional annotation is to identify, because we have actually interpreted the conserved region, that where was that particular amino acid present, but now we have to identify what is the function of that particular protein. So for that we need to search for that protein in the NCBI conserved domain section, and there we get to know that in conserved domain database analysis, it was revealed that there is a presence of a DECP HTRA domain which spans from 44 to 461 amino acids with a highly significant E value, indicating that it's a complete and functional protein. So, in conserved regions, we could not infer that our proteins amino acids, which one of our proteins amino acids are functional. So in this conserved domain section we have finally understood that our amino acids ranging from 44 to 461 are actually responsible for the protein's function. So for interpro analysis, what we did was that we understand that what type of domains are actually responsible for the work. So 466 amino acid protein belongs to a serine protease family, and it contains multiple domains, two PDZ and one trypsin-like serine protease domain. So for signal P, we understood that actually what is happening inside the 466 amino acid. Just a moment, ma'am. बेटा आप थोड़ी देर यहाँ से चले जाओ. Am I audible?
Yes, you are audible.
Okay, so as I was saying that in this signal P, what the red line is actually showing, it's like the first 22 amino acids that act as a signal peptide. So signal peptide is then cleaved off, and then in this green portion, we can see that after the cleavage of 22 amino acids that act as a signal peptide, the mature protein amino acids are shown. And this SEC dependent pathway actually represents that the protein which is formed is mature and it is not going to function in the cytoplasmic region. To confirm our analysis further, we go for P-sort-B analysis, which actually confirms the location where amino acids are going to, where our amino acid like the TSA47 is going to function. So as we can see that after performing P-sort B, we realize that it is in the periplasmic region. Okay, so in the smart section, we actually analyzed that what are the homologs with this protein. Trypsin 2 domain indicate that protein has a serine protease function. Two PTZ domains were detected. The first with high confidence, ranging from amino acid 269 till 337, and the second amino acid 364 with lower confidence, as 364 to 454 with lower confidence. Collectively, the mature protein, it appeared to be a functional protease domain and protein interaction modules. So the string is also done to identify the interaction of the TSA47 protein with other proteins, which actually predicted that the predicted partners along with TSA47 protein is histidine kinases, and this interaction actually shows the very first slide of the functional annotation, which was saying that the function of TSA47 is in stress responsive signal transduction pathways. That's it. Now Sidesh will recontinue, ma'am. Thank you.
Uh, s- so, Samin, if you stop sharing, then I can.
Okay.
Okay. Hope it's visible.
Yes.
So, I will continue with the step 6, which was biological interpretation. The step was performed by Vaidehi. So, in the biological interpretation, the key findings were that the protein belongs to HTRA or DECP family of serine proteases. To just summarize this particular protein, I also did a little bit of search, and I saw that the DECP family of proteases are also present in many of the gram-negative bacteria, which are pathogenic in nature for humans. So this is very crucial protein, it seems, for these bacteria. It was predicted to be a periplasmic protein and not a cytoplasmic one. Although I would like to mention here that it is a huge debate whether this protein is a secreted one or periplasmic protein. In some bacteria, depending on the conditions, it can be both. The domain structure showed that the protein is complete and functional, and overall it was seen to be biologically active in the bacteria. Now, what was the predicted role of TSA47? It acts as serine protease. Serine protease are specific family of proteases which are responsible to cleave amino acids in the hosts, and these are also known to be present in other microorganisms such as viruses, wherein they are actually present in the host and cleave the proteins. It helps in removing the misfolded and damaged proteins. So it acts as a quality control protein in case the bacteria has to survive in adverse conditions. It also supports proper folding and maintenance of exported proteins, and which is why it helps the bacteria to survive under stressful conditions. Now, what is the biological and pathogenic importance? It helps the bacteria to survive inside the host naturally, because Orientia Sutsugamushi, I would like to also say that it is very slow growing bacterium. So the doubling time of the bacterium is 9 hours or 9.5 hours. So it is very slow to grow, but which is also why it needs this to survive inside the host, which is humans. Stress response proteins are important for its survival during infection, and the protein helps to maintain the stability, as we saw in the earlier slide. Proper protein maintenance supports bacterial growth and its persistence in the host. So to conclude, based on all the in silico analysis, the protein was predicted to function as a periplasmic HTRA-like serine protease, and it is known to be involved in maintaining the quality of the proteins and to manage the stress which occurs for the bacteria while it is duplicating inside the human host. These functions support the bacterial survival during infection, which is very logical, And TSA47 has been also experimentally shown to share homology with human HTRA1. So this was a very interesting paper that I also saw, and I thought I should mention it here, indicating the conserved structural and functional features between bacterial and human serine proteases. So the identity was around, if I remember correctly, between 46% to 56% in a specific region of the serine protease. So it's very clear that it indeed is a serine protease, and it has its proper biological function. With that, I would like to acknowledge and thank Muskan ma'am here, who trained us very well. Thank you very much, ma'am, for the training as well as for all the dedicated time and doubt solving. And also all the team members, Gayatri, Paripurna, Purvi, Samin, and Vaidehi, who took all the efforts and time to make the project happen in the group together. With that, I would like to end the presentation. Thank you.
Okay, that was some really nice work by your group, especially the functional annotation part. That was a really knowledgeable part. I really liked that one. So how
Thank you,
much
ma'am.
time did you guys take to complete this presentation?
Ma'am, to be very frank, I think we were working on it since like yesterday and today. So no, I mean day before yesterday. Yesterday, yeah.
So it took you 3 days to complete the project?
No, no, ma'am. I think
No,
it was
no,
like
like only
a
the
week.
presentation part.
Okay, okay.
Yes, ma'am. We have been working on our, the specific parts for the past week or more, I think. But
Yeah.
the presentation part is like done within the past 2 days.
No, I'm really glad that you did the presentation as it helps you understand your own project as well. As well, it helps you to understand more things that you didn't even learn in the course. So it helps you to research yourself for some better knowledgeable things. So I really liked your presentation.
Thank you, ma'am.
Okay, I will just ask in group if somebody else want to do the presentation.
Ma'am, I have a doubt.
Yes, Gayatri.
Ma'am, so when we were doing the quality analysis, I couldn't crack the code for like how to filter the C terminal and N terminal bases, ma'am, the first 20 bases.
Okay. Can you share
I
me
have,
the code?
yes, ma'am. Ma'am, can you just give me like a couple of minutes, ma'am?
Yes, okay.
Ma'am, just one doubt I wanted to ask. Sometimes the code when it was run, it shows like it's taking longer than 10 minutes or something. Please rerun or something like this. So I was just wondering, is it like low RAM issue or like the code itself was not proper? But if the code was not proper, it wouldn't run in the first place, right?
Hmm. Maybe the sequence you take was very long. It depends on that as well.
Ma'am,
But
I
ma'am, it was
tried doing
like
both.
1401.
Ma'am, I tried doing both. Initially, my blast was also running,
Hmm.
but then it was like 466 amino right? But for the very long sequence, it was taking more than 10 minutes, and it was giving a warning or error issue that it is taking more than
Yes,
10 minutes.
for
But when
longer
I tried with
sequence, it takes time.
yeah, but I also tried with shorter sequence, but it didn't run. The terminal was empty. I don't know what went wrong. Maybe it was a glitch.
Blast usually takes 5 minutes if you have a shorter sequence, but you have a longer sequence, it takes more time.
Okay.
Okay, I don't think there is anyone else who wants to do presentation today. Okay, let me stop sharing the screen. Stop the recording. Wait a minute.